Assalamu alaikum everyone. This is the third episode of the microbiology series, and I'm your host Faris Al-Shboul. In this episode, we will start the chapter 2, the microscopes, and then in the next episode we will finish chapter 2. So we'll start this chapter by discussing the general sizes of microorganisms. So in general, we have the algae as the biggest, then the protozoa, then the fungi, then the bacteria, and the smallest are the viruses. All microbes are measured by micrometer, which is 10 to the power of -6 meters, 1 part in a million meters, except viruses, which are measured by nanometer, 10 to the power of -9 meter, 1 part in a billion parts of meter. These are the notable sizes that you have to memorize. Most viruses measure from 20 to 300 nanometers in diameter. The spherical bacteria, the cocci bacteria, has a diameter of 1 micrometer on average, while the rod-shaped bacteria, the bacilli bacteria, measure 1 by 3 micrometer. The fungi, the yeast fungi, has a diameter of 3 to 5 micrometer, while the mold fungi, their structure, the hyphae, has a diameter of 3 to 30 micrometer. The aseptate hyphae is bigger than the septate hyphae in diameter. Lastly, we have the parasitic protozoa, which measures below 50 micrometer, while the free protozoa above 50 micrometer. The difference between parasitic and free: the parasitic protozoa, they need a host, a living creature to infect and cause disease and to replicate inside that. While the free protozoa, most of them live in pond water, miyah al-bukhayra, and they don't need to infect anything. We have the 4 types of microscopes: the simple microscopes, the compound light microscopes, the electron microscopes, the scanning and the transmission electron microscopes, and lastly we have the atomic force microscopes. As a general information, simple microscopes are called simple because they have one magnifying lens. Compound microscopes, they are called compound because they have multiple magnifying lens, and they have light as the main source of illumination. Electron microscopes are called electron because they use electron beam as the source of illumination. And lastly, atomic force microscopes use the atomic forces to scan specimens. All of these are discussed in further details in the next episode. Now we will discuss some microscopic terms used in microscopy. The image taken by compound light microscopes is called a photomicrograph. The image taken by electron microscope is called electron micrograph. So we have the photomicrograph and the electron micrograph, the scanning electron micrograph and the transmission electron micrograph. Okay, moving on to the most important term in microscopy, which is the resolution or the resolving power. We all hear about resolution in our cameras, in our smartphones. So what is resolution? Resolution is the distance between the closest 2 points in the image that you can see them distinct and separate. The smaller the resolution number, the better. Now, resolution reflects how clear is the image, how much detail you can see in that image. So the more dots you see in an image, the more pixels you see in an image, the more details you can see in it. So we might have a 2 megapixel camera and we might have a 10 megapixel camera. The 10 megapixel camera is obviously better. Why? Because there is more points in a single photo than the previous one, the 2 megapixel. So resolution describe the distance between any 2 points that you can see them distinct. Okay, so this is a slide that I made to furthermore explain what is resolution. So we have the petri dish right here, Petri dish is a glass, a flat glass that is used to grow bacteria. Each dot you see in the glass is a bacterial colony having many hundreds or thousands or even thousands of bacteria. So by our naked eye, we can see each colony as one dot, right? So if we magnify this dot over here, the naked eye see it as one dot, while in fact, in reality, it is made of multiple dots, each dot representing a bacteria. So if we have a better resolution, and this magnification will reveal more dots, right? But the naked eye can't resolve more dots. So the compound light microscope can do that. The compound light microscope has a resolution of 0.2 micrometer, while the naked eye has a resolution of 0.2 millimeter. So what are these numbers? These numbers reflect the distance at which the naked eye or the microscope can make a distinction between any 2 points. Below this threshold, below this number, the naked eye or the microscope cannot make the distinction. It will merge all the points as one point, or even cannot see below that limit. Okay, if we take each bacteria and have a better resolution using the transmission electron microscope, which has a resolution of 0.2 nanometer, we will see even more details inside each dot, which is each bacteria. So each bacteria has the genetic material and the plasma membrane, etc. So using the transmission electron microscope, we can see all that. This is due to the small resolution, resolution number. The electron microscope can see images in the nano world. Okay, so the resolving power depends on the wavelength that is being used to view the micrograph. The smaller the wavelength, the better the resolving power. So visible light has a wavelength on average of 0.45 micrometer, while the electron has a wavelength of around 1 nanometer. So that is why the electron microscopy can see more detail, because it has a wavelength that is much smaller. Okay, this is the equation that you have to know, or you have to think about. Resolution equals wavelength lambda over 2 numerical aperture. The maximum resolution is half the wavelength. So that's why the compound light microscopes has a resolution of 0.2 micrometer, which is around half the wavelength of visible light. About numerical aperture, what you have to know, numerical aperture is the light gathering ability of the microscope. This is dependent on the lenses used in that microscope, the refractive power or the focusing power of that lens. We can increase that power by using oil immersion or even water. Air has the lowest refractive power, then the water more, and the highest is oil immersion, which increases the refractive power of the lens. Also, numerical aperture depends on the size of acceptance cone of the objective lens. This is a topic on optics, and you don't have to understand everything. But the concept here, the closer the objective lens to the specimen, the higher the numerical aperture, because anything that is more closer, for example, to your eyes, you can see more details. When it is more farther away, you can't see much. So the closer the objective lens to the specimen, the higher the numerical aperture. Okay, so this is a summary of what we just said. Resolving power of the naked eye is 0.2 millimeter. Resolving power of all compound microscopes is 0.2 micrometer. The resolving power of scanning electron microscope is 20 nanometer, while the best resolving power is of transmission electron microscope, which is 0.2 nanometer. Okay, now a new term, which is contrast. Contrast is the difference in light intensity between the image and its background. So, have a look on this image and compare it to this image. These are two micrographs taken by compound light microscopes. This is taken by one type, and this is by another type. So you can see this image, you can't differentiate that much between the specimen and its background. We say this has poor contrast because the eye needs a contrast of at least 2% in order to distinguish the differences between the image and its background. The poor contrast because the colors are so similar that the eye cannot distinguish them apart. While here in this micrograph, we have a good contrast, even though both of these are taken by the same, let's say, general type of microscopes, compound light microscopes, but this one has a good contrast, while this one has poor contrast. Magnification power. Magnification, we all know what it is. Magnification power is the maximum power to magnify objects, the most important thing, without they get blurred. So if you can magnify a picture without it being getting blurred, this is magnification power, true magnification power. While at the moment it gets blurred, this is fake magnification. We call it empty magnification power. So it's fake magnification that image becomes blurred and unrecognizable because of the resolution limit. Magnification power of simple microscopes, these single lenses, is usually between 3 to 20 times. Leeuwenhoek was expert in making magnifying lenses, and he was able to make a simple microscope that had 300 times magnification power. Now the magnification power of all compound light microscopes is up to 1000 times. The magnification power of a scanning electron microscope is 10,000 times. The magnification power of transmission electron microscope is 200,000 times. This is the episode that we will start in the next episode. Thank you for listening and be tuned. Assalamu alaikum.